Welcome back to our video lecture series dealing with the cell fate. After we have a quick look how cell death or apoptosis is regulated, let us have a look how proliferation or cell division occurs. I would like to start this video with a video showing you dividing cells in the dish. As you can see here, even in the dish, if we cultivate the cells in presence of certain signal, cell division can take place. And this is quite regulated process, because if cells divide in an uncontrolled manner, we would end up with disorders or conditions like cancer. So obviously this process must be fairly tightly regulated. And even in the dish, not all of the cells are dividing, even though they receive signals for cell division. So, in order to understand proliferation or cell division, we need to have a look at two underlying processes, namely at the cell growth and cell division, because those processes are actually not the same. Cell growth involves an increase in cell size without an increase in cell number. So our cells grow because there is an ongoing synthesis of new proteins, membrane lipids, and other parts of the cell that are required to produce the other cells. It involves increase of the number of organelles, and also may involve replication of the DNA, but this is actually not always necessary. The cell growth per se is controlled by different signals, and those include growth factors and also contact with other cells. Contact of a cell with other cells usually controls cell growth negatively, and this is something you could have seen in the video I showed you before. Not all of the cells were dividing, and those dividing cells actually did have a bit of space in the cell culture dish, allowing them to divide. So the next process involved in cell division is cell proliferation. It always involves replication of the DNA and also division to produce two daughter cells, and this is something we had a closer look at in our lecture dealing with mitosis and meiosis. Proliferation usually follows on from the cell growth phase, and this makes perfectly sense, because both daughter cells must be identical. They need to have all of the organelles required in a cell, and also all the genetic material of the parental cell. If not a reductive division would occur, such as in case of the meiosis, the result of proliferation is one or more smaller cells than the parent cell, because again proliferation follows on the growth phase. If we look at the regulation of proliferation, then we will notice that they are both positive and negative signals. The positive signals usually involve growth factors. Those growth factors bind to the respective receptors. This results in a conformational change of the receptor, phosphorylation of the receptor, and activation of data proteins. And here we must come back to the Ras protein I showed you before, because Ras is extremely important in health and disease. Ras is usually involved in cancer cells. If it is hyperactive or mutated, Ras activity does no longer require binding of the growth factors. Therefore, cancer cells divide without sensing those factors and proliferate in an uncontrolled manner. In addition to positive growth factors, there are also growth inhibiting factors. If those factors bind to the receptor, this activates transcription factors that regulate cell division negatively. And a very nice example of such transcription factor is P53. In addition to RAS, also p53 is quite commonly involved in cancer development, because if it is mutated, it can no longer negatively regulate cell growth and cell proliferation. So, in order to fully understand proliferation and the cell cycle and cell division, we need to have a look at the cell cycle. And this is something I showed you fairly briefly in our mitosis lecture. So, as you might remember, mitosis is only one phase of the cell cycle, actually the shortest one. Mitosis involves division of the cell and cytokinesis, where two cells are produced from one daughter cell. However, the cell cycle, which is in human cells usually around about 24 hours long, also includes other phases, including the G1 phase, also referred to as gap 1 phase, the S phase, where DNA synthesis takes place, and G2 phase, or gap 2 phase as well. If a cell divides, the cell cycle needs to happen. However, the transition between each of those cell cycle phases is fairly tightly controlled. And here we are talking about so-called cell cycle checkpoints. Those checkpoints block the cell cycle and have to be actively removed by the cell to allow the cell cycle to proceed. And the first of those transition points or checkpoints is actually at the end of the gap 1 phase. Here the cell checks if the environment is favorable for cell division. Are the growth factors present? Yes or no? Are death factors present? Yes or no? If there are no death factors, survival factors are present, and also growth factors are sensed by the cell, then the cell can proceed into the S phase and enters the cell cycle and starts to synthesize DNA. The next checkpoint within the cell cycle is the G2M checkpoint. Here, the cell checks if DNA is replicated correctly. This is also quite important checkpoint, which is no longer under control in cancer cells. And this is something quite fascinating. As you might remember, in normal cells under physiological conditions, the number of chromosomes is quite defined. And we do have to look at 46 chromosomes in human cells, or 22 pairs of autosomes and 2 sex chromosomes. In cancer cells, the chromosomal configuration is no longer diploid. Research has revealed in the last decade or so that the usual typical chromosomal configuration of cancer cells is round about 70, because this checkpoint is no longer working properly. In addition to checking the DNA, the cell double checks again if the environment is favorable. If this is the case, the cell is able to enter mitosis. The last checkpoint is the metaphase to anaphase transition. Here, the cell checks if all chromosomes are properly attached to the spindle, and this triggers anaphase, and then the cell is able to proceed to cytokinesis, resulting in 2 healthy daughter cells. All of those transition points or checkpoints are tightly controlled by very specific signal transduction pathways. Activation of the signal transduction pathways usually removes the cell cycle roadblocks. So again, cell cycle and proliferation need to be actively regulated. The next possible cell fate we will look at is the process of differentiation. And differentiation is something extremely important during development, where we start with uncommitted embryonic stem cells, but also for adult stem cells present in healthy adult individuals. Stem cells are uncommitted cells that can differentiate or specialize into many different cell factors. Stem cells, especially embryonic stem cells, have a very high differentiation potentials. Embryonic stem cells can give rise to literally any cell type in your body. They can generate neurons, bone cells, blood cells, and so on. In contrast, adult stem cells have slightly more limited differentiation potential. However, they are still able to generate quite a lot of different cell types. And we will have a look at stem cells in one of our dedicated lectures in the spring term. So, differentiation is the process by which stem cells become specialized cell types. Usually, but not always, this process involves them ceasing to proliferate. And what I would like to show you now is differentiation of embryonic stem cells into nerve cells in a dish. Undifferentiated embryonic stem cells are quite small and they appear fairly roundish, and each of those circles represents one stem cell. If we now artificially introduce cell division or differentiation signals into the cell culture, this results in a quite rapid and actually quite dramatic change of the shape. As you can see here, those cells become more elongated, several vesicles are getting secreted. What you can see here is appearance of first bipolar cells, then neurids formed, and this process can be fairly rapid in case of embryonic stem cells. This is slightly slower in adult stem cells, but still fairly efficient. In this video, which is actually a video taken in my own lab, you will see a layer of embryonic stem cells exposed to certain signals for 1 week. And this cluster in the middle has been differentiated into beating cardiomyocytes, into beating heart cells. So this differentiation process can not only take place in the body, but also in a dish. And this is extremely important for the pharmaceutical industry, because now we can differentiate embryonic stem cells in a dish into heart cells and test different drugs that are designed to treat cardiovascular conditions. So obviously, differentiation is quite a complicated process. And similar to cell proliferation, it is tightly controlled by different signal transduction pathways that usually activated by ligands that lead to expression, activation of promoter-specific transcription factors. As we have learned before, transcription factors bind to regulatory sequences on the DNA. They recognize target sequences in the DNA. And there are two classes of transcription factors. We have general and promoter-specific transcription factors. General transcription factors are active in most cell types and are required for transcription. In contrast, promoter-specific transcription factors can influence transcription positively and negatively and in a cell type specific manner. Different sets of promoter-specific transcription factors are active in neural cells. We have different sets active in blood cells, in muscle cells, and so on. This is actually quite important for specific gene expression in every cell type. So we have tissue-specific expression of those promoter-specific transcription factors. They can also be expressed at a specific time during the development. And this is important for development. An example I would like to show you is embryonic muscle development. What you can see here is an embryonic stem cell or embryonic precursor cell. Here, certain genes are off. Here we are looking at differentiation of embryonic stem cells into muscle cells. So we have master control genes which are in charge of expression of certain muscle-specific genes. So if the master control gene is off, other muscle-specific genes are off as well. If the embryonic stem cell now receives receives a signal leading to expression of the transcription factor called MyoD. Then this master control gene is expressed. In turn, mRNA is produced, MyoD protein is expressed, and this in turn activates other muscle-specific genes. So we have a tightly controlled signal transduction cascade which is in charge of differentiation of the cell. Once those tissue-specific or muscle-specific genes are on, we have differentiation of the cells into fully functional muscle cells. In contrast to positive activation of differentiation specific genes, differentiation can also be activated in a negative manner, and this is the case in embryonic stem cells. Embryonic stem cells are kept in an undifferentiated state by expression of certain transcription factors, including the most important one called OCT4 and NANOG. So, if OCT4, NANOG are expressed, this usually inhibits activation of differentiation-specific transcription factors. Therefore, the cell is kept in an undifferentiated state by those transcription factors. If they are no longer present, differentiation-specific transcription factors can be activated and the cell can specialize into different cell types. With this, I would like to end session number 2, and I will see you in session number 3. Thank you very much.